Hi everyone, we're group 7 and we'll be presenting Campylobacter bacteria.
What is Campylobacter bacteria? Campylobacter bacteria is a zoonosis-type pathogen that causes the disease from either animal organs and byproducts or directly from animals to humans. It's an S or curved broad-shaped pathogen. It totally includes 17 species with 6 subspecies and is one of the 4 major global causes of diarrheal disease and the most common bacterial cause of gastroenteritis worldwide. Annually, it infects upwards of 1.5 million Americans and 96 million people worldwide. How it is spread. It is spread through animals such as cows and chickens, and it often occurs through consumption of uncooked poultry, unpasteurized milk, and untreated water.
Immune response impact. Campylobacter bacterial infections typically cause gastritis in humans. Symptoms include diarrhea, abdominal cramping, fever, nausea, and vomiting. Upon ingestion, the bacteria invade the intestinal lining, leading to inflammation and an immune reaction. The release of inflammatory mediators combined with the damage caused by the bacteria results in the symptoms previously mentioned. The body recognizes Campylobacter as a threat, leading to the activation of various components like the innate and adaptive immune system, attempting to eliminate the bacteria. Diarrhea is often a result of the body's attempt to flush out the invading pathogens. The severity of symptoms can vary, and in some cases complications such as Guillain-Barré syndrome is a rare neurological disorder where the body's immune system mistakenly attacks the peripheral nerves. It often follows an infection leading to symptoms such as muscle weakness, tingling, and in severe cases, paralysis. The exact cause is not fully understood, but it is thought to involve an abnormal immune response triggered by infections with Campylobacter jejuni, being a common associated bacterium. While most individuals with GBS recover with supportive care and treatment, the condition can be serious and may require medical intervention, such as intravenous immunoglobulin therapy. Early diagnosis is crucial for a better outcome in managing this potentially life-threatening condition.
What cells and tissues is it in? Campylobacter resides inside the mucosal layer of the gastrointestinal tract. It is inside the single layer of intestinal epithelial cells, IECs, that make up the mucosal epithelium of the small intestine. The flagella is used as a key adhesion for this pathogen. Once this pathogen is ingested, it binds to the mucosal tissue where it takes on its gastrointestinal pathology. Once the pathogen makes its way intracellular, translocation of the Campylobacter begins within the epithelial cells.
What are the first cells of the immune system to encounter it? Neutrophils are the first immune cells that contact the Campylobacter infection. When the PRRs on the surface of the intestinal epithelial cell bind to the bacterial pens, they send out cytokines attracting the neutrophils. Intestinal epithelial cells produce IL-8, a neutrophil chemoattractant factor that induces neutrophils into the intestinal submucosa where they infiltrate the site of inflammation. So the neutrophils have 3 main mechanisms: phagocytosis, degranulation of proteins found in the microbes, and formation of the neutrophil extracellular tract, known as nets. During the degranulation process, the neutrophils release little cytotoxic particles in their cells, known as granules, to break down the proteins in the bacteria, making it easier to phagocytose. The actual process of phagocytosis is when the neutrophil attaches itself to the bacteria, engulfs and internalizes it, and digests it, then eliminating it. With these mechanisms, the neutrophils degranulate the pathogen before phagocytosing it, and the bits of bacterium that escapes are trapped into the net.
What signals do they send to the rest of the immune system and how? The neutrophils release IL-6, TNF-alpha, and IL-1 beta cytokines during the degranulation process, leading to recruitment of other innate immune response cells, such as NK cells, dendritic cells, and macrophages. These cells are recruited to help to accelerate the phagocytic process. Through chemokine and cytokine signaling, these cells interact with neutrophils and aid in the breakdown of pathogens. When macrophages and dendritic cells are recruited to the site of infection, they perform both inflammatory and anti-inflammatory signaling. Once the neutrophils have infiltrated the submucosal tissue to the site of infection, they present a CD11b cell surface marker. This surface marker initiates the phagocytic process conducted by the macrophages and monocytes. Neutrophils also release ROS, which are toxic radicals that are oxidative forms of killing to kill the bacteria. When neutrophils produce ROS and the other inflammatory cytokines, it damages the crypts, which is the pocket of epithelial cells inside the intestine. This is what causes the symptom of diarrhea experienced during the infection.
What other immune cells become involved? In later stages of infection, T and B cells, also known as lymphocytes, become involved in fighting the infection. With the help of innate immunity, lymphocytes recognize and memorize the unique antigen of a foreign material or pathogen and develop an antigen-specific response to eliminate it. This happens with the help of APCs, GCs, macrophages, and monocytes, which bridge the gap between the innate and adaptive immune system by presenting the antigens found in the bacteria to the T cell and B cell with their corresponding PCR and BCR. T cells, when activated with the help of GCs, become helper T cells. The helper T cells are Th1 and Th17. The Th1 cell combats the bacteria that have made their way intracellularly by activating macrophages to help with the killing process through phagocytosing the bacteria-infected cell. While the Th1 cells do that, the Th17 cells combat the bacteria that's still extracellular in the barrier tissues. For the B cells, they get activated with the help of Th1 cells providing the CD40 ligand needed for activation. When mature, they begin to produce IgG, IgA, and IgM antibodies, which then go back to the point of infection to fight the bacteria.
The infection mechanism of Campylobacter is composed of mainly 4 stages: adhesion, colonization, invasion, and toxin production. During adhesion, it will adhere to the cell surfaces and is able to invade the mucosal layer of the intestine and colon. By this, it reaches the intestinal epithelial cells. And at this point, it will colonize the entire digestive tract. During invasion, Campylobacter is able to secrete CDT toxin proteins, which will cause cell cycle, cellular arrest, cell swelling, or apoptosis, and it will release Cas9 nuclease, which causes DNA damage or apoptosis. The CDT toxins disrupt the epithelial barrier and impair signaling pathways of the immune system. It also releases Campylobacter invasion antigen proteins that stimulate TLRs and induce P3 mitogen activated protein kinase and extracellular regulated kinase pathways. This drives IL-8 secretion. This will result in the neutrophil chemotaxis to the site of the infection.
After the innate immune system attack on the bacteria, macrophages and dendritic cells take antigen presenting markers from the bacteria and present them to naive T cells and B cells. This creates maturation of T cells into Th1 and Th17 cells that produce cytokines to help colonize apoptosis. The B cells go on to become mature B cells, as in memory cells and plasma cells, which will then secrete antibodies against the infection.
Resolvement of infection. Does it get cleared or resolved? Yes. In many cases, a Campylobacter infection is resolved as the immune system successfully clears the bacteria from the body. The immune response, involving various components such as neutrophils, macrophages, and T cells, work to eliminate the Campylobacter pathogens. Additionally, the body's natural defenses, including the production of antibodies, contribute to the clearance of the infection. Most individuals with Campylobacter gastronitis recover fully without specific medical treatment. However, although the symptoms can be resolved on their own, the duration and severity of these symptoms can vary, and in some cases, antibiotics may be prescribed, especially for severe or prolonged infections. It's important for individuals with symptoms of Campylobacter infection, such as diarrhea and abdominal cramping, to stay hydrated and seek medical attention if symptoms are severe or persistent. Overall, the majority of Campylobacter infections resolve on their own with the help of the immune system and appropriate supportive care.
Now, because Campylobacter may be contracted waterborne or through the consumption of poultry and meat products, there are many global and social economic impacts. For some of the global impacts in low-income nations, various environmental sources such as streams and rivers where people depend on these bodies of water as their major drinking water source, it increases their chance of infection. In high-income countries, water is found contaminated through faculty pumps and pipes, and the most high-risk population is farmers. As for social and economic impacts, there's an increase in investment in sewage sanitary conditions and protocols, and an increase of public awareness and education. Now, for funding, the majority of funding is done through the government and Food and Drug Administration, and funding and research. So they can prevent outbreaks that are both foodborne and waterborne. In low-income areas, they benefit from funding and studies of the bacteria as it aids their knowledge of consuming clean water and pushes for addressing the public health issues. In high-income areas, it benefits as it allows for an increase in awareness of city management, which will increase the knowledge and action of bettering water systems and regular checkups.
Although the Campylobacter has been heavily researched, they have found that it's very difficult to phenotype them, leading for newer ways and methods that we can ease the process. The disease also can create long-term bowel issues and gastrointestinal issues, and prevention of that during infection and after infection can also be researched. Campylobacter has also remained a hard species to study because of its low metabolic rate and time-consuming studies, so further research into how we can create an optimal growing environment can be advantageous.
Prognosis of Campylobacter. The prognosis for Campylobacter infections is generally favorable. Most cases resolve on their own without specific medical treatment. The duration of symptoms can vary, but individuals typically recover fully within a week to 10 days. Now, if you are to find yourself with a Campylobacter infection, self-care is necessary. Supportive care is the primary approach, focusing on maintaining hydration and alleviating symptoms. Adequate fluid intake is crucial to prevent dehydration, especially in cases with diarrhea. And finally, rest is recommended to aid the body's natural healing process. Now, if your symptoms are to worsen, there is always antibiotic care. Antibiotics are generally reserved for severe or prolonged cases, immunocompromised individuals, or those at risk of complications. If prescribed, antibiotics like azithromycin or erythromycin are commonly used. With that said, you can also have complications. While rare complications like Guillain-Barré syndrome may occur, necessitating close medical monitoring, individuals with severe or persistent symptoms, bloody stools, high fevers, or signs of dehydration should seek prompt medical attention. Some preventative measures are practicing good hygiene, including through hand washing and safe food handling, helps prevent Campylobacter infections. Also, ensuring poultry and other meats are properly cooked can reduce the risk of infection. And here are our citations.
Thank you for listening to our presentation.